Okay, it's 5 after, so I'm going to go ahead and start. Thanks for coming out at the unusual time and unusual place for the EEG, fMRI, and language talk. I've redone this a little bit since last week, or last year. So last year I made it a lot more about simultaneous methods and language, and this year I'm going to make it a little bit more heavy on EEG, ERP methods. I'm still going to talk about sort of the converging evidence of a reading circuit between EEG, MEG, and fMRI. And then I'm going to highlight sort of the importance of simultaneous EEG, fMRI studies moving forward, of which we have the equipment here at NIH. So if you're interested in doing simultaneous data collection, come talk to me. I seem to have landed in the position of helping people get their studies up and running. So unlike every other neuroimaging technique, EEG actually has a very, very nice history. Some of the earliest experiments were conducted by Hans Berger in 1924, but you can imagine that if you just kind of look up here, that literally you could have someone sitting with a single electrode or two electrodes attached to them and their foot in a bucket in order to ground them, and somehow record something that resembled brain activity. Now we know from this and also many, many other studies that you never want to ground your subject like this, because should you run into an electrical malfunction, that would mean that the electrical current would run through the equipment and then through your participants and then through the ground, and that's usually bad. You don't want your participants to experience pain whenever you have technical malfunctions. But nonetheless, EEG sort of developed from single electrode recording to multi-electrode recording. This is a picture of a whole bunch of amplifiers so that we could collect, say, up to 16 channels. And here's a grass amplifier cart, which I think is 8 channels. And now today we have a system here at NIH that will collect up to 256 electrodes. So you can imagine that we started with single electrodes that took up almost an entire room to, say, 16 channels that took up an entire room to 8 channels that took up a cart to now we're at 256 channels that basically are the size of my laptop. Much more portable than fMRI. Of course, the major trade-off for EEG compared to other neuroimaging techniques is the temporal spatial resolution. So you can see that MEG and ERP tend to have fairly fast temporal resolution, so down to the millisecond, but we sacrifice a little bit of our spatial resolution, whereas, say, fMRI may come down slightly lower to the column or even the layer once we get to 7 Tesla. If you saw Renzo Huber's talk on Monday, or if you're going to see it again, or one of his other talks on Friday, he'll be talking about that. Nevertheless, MEG and ERP are great for giving us indicators at the millisecond of what the brain is doing, instead of having to wait for these secondary indicators like the bulge response, the blood flow, responding to neuronal activation. And in general, it works like this. So we get a series of neurons that fire together. We oftentimes refer to these as dipoles. These are mostly composed of pyramid cells in the brain. And this sort of activation of the brain or electrical discharge produces a particular scalp distribution of electrical currents. This makes great art, by the way, if you're looking for something to hang. We can then record these electrical currents using EEG electrodes over the head. So this is 128 electrode nets. And then those get represented as lines showing microvolt changes from 0. So another important change of EEG and MEG is that we actually have a baseline measure, which is 0 volts or 0 microvolts, whereas in fMRI we sort of have to build a baseline from all the other activation or activity. So we have real units. EEG also reflects different types of brain states. So if we just put a few EEG electrodes on anyone in this room, you probably came in here fairly excited. And now that you're hearing me talk, you're sort of relaxing. As we get towards maybe like 45 minutes in, everyone's going to get a little bit drowsy. Hopefully I'll throw in enough jokes that no one falls asleep, and hopefully no one goes into a deep sleep or even a coma state. But raise your hand if you get there, because we'd love to study someone who can raise their hand while in a coma. Nonetheless, if for no other reason, EEG is great for monitoring the state of your participants. Say you could even do this in the scanner to find out if people are asleep, which is, trust me, always a challenge. In terms of going from the EEG to the evoked potential or event-related potential, we have this continuous ongoing EEG signal, and these little S's represent different stimuli being presented. We just grab a section of time around each of these stimuli, say about a second. We average them together, and we get a series of peaks and valleys that I'll go into in a second. So as I mentioned, this is called the event-related potential, or ERP, never ERP, ERP. It's a portion of the ongoing EEG. It's time-locked to stimulus onset. It gives us strong temporal information, and it's comparable across the lifespan. And I'll just say that the way that we sort of differentiate the different peaks is that we either count them and their polarity. So say the first positive peak is our P1, the first negative peak is our N1, P2, N2, so on and so forth. Or we give them the millisecond value. So this may be the P70, this may be the N100, the P200, so on and so forth. In about 2000, Picton led a group of researchers to come up with an international standard that was to say everyone should use the millisecond value, but nonetheless you'll still see plenty of people identifying sort of the rank order of the peaks. In 1979, Manny Donchen came up with this definition of ERPs that I feel is required to sort of read aloud. So the ERP is a sequence of overlapping components, each perhaps representing activity of different populations of nerve cells, and each sometimes standing in different, perhaps orthogonal, relations to experimental variables. It's just sort of an picture to show you that ERPs are very comparable across the lifespan. So this is a young baby who's now, I think he's 27 years old today, or thereabouts. And when we talk about comparability across the lifespan, you can actually see that the latency of that first negative peak, the N1, decreases as you get older, but it decreases with different slopes depending on, say, your verbal IQ. Not all peaks have sort of this linear effect. The P2 is an example where we have sort of this curvilinear property. And this was work from Kim Espy that came out of Southern Illinois University while she was there. Most people can immediately recognize that over time, ERPs tend to start small in newborns and grow larger until you hit about 8 to 10 years of age, and then they tend to get smaller again and slightly faster. And just because some people don't believe me in terms of the being able to do this with babies, this is a baby that is about 12 hours old. So still in the nursery, in the hospital. We kind of sneak in with permission, but we sneak in because the nursing staff is often very busy. And the very first thing that we do is measure the circumference of the head. This allows us to identify what size net we should put on the baby. Then we measure from the nasion to the enion, that little bump in the back of your head, and we put this designer mark in the center of the head so that we know exactly where, with washable marker, but so that we know where the center of the net should go. The net's been soaking in potassium chloride, which is basically just fancy salt water, and goes on in the matter of a few seconds. Adjustments take on babies about 5 minutes. On adults, usually we can get 256 electrodes onto an adult in about 10 minutes. The baby often usually wakes up for this and then immediately falls back asleep. So put the net on, put the baby back in the bassinet, and the next thing that we do is we actually measure the impedance, or the amount of resistance each electrode is detecting, and then measure EEG to different speech sounds in this case. Take off the net, dry off the baby's head, sometimes change the diaper, and then quickly rush off to the next baby in the next bassinet so that we can stay one step ahead of the nursing staff. Okay, I've already explained the nomenclature in terms of P1, N1, or P70, N100, but I just want to draw your attention to this idea that we have exogenous variables and endogenous variables. So exogenous tend to be the peaks that happen earlier in time, and they are thought to directly represent the physical characteristics of a stimulus. And this is fairly easy to show in terms of the brightness of a stimulus or the loudness of a stimulus. So if you play different sort of increasing loudness of tones, you see that the N100 gets larger over with the loudness of the tone. The endogenous peaks are thought to be something about internal processing. So these don't modulate with the stimulus characteristics in terms of loudness or brightness, but more with the sort of cognitive processing that may happen afterwards. So the P300, for instance, is, or one of the P300s, there are many, is modulated by an oddball response. So if you're playing a consistent tone of beep, 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 boop, whenever you hear the one that's odd, the P300 sort of rears its larger amplitude in response. That was Sam Sutton, and then Marta Kutas, of course, is famous for the N4 or N400 being related to sentence processing. I'm not going to go through all of these because you can get the slides online, but this is just to give you an example of all of these peaks have been fairly well characterized over the years, and you see that there's slightly different latencies in terms of auditory and visual. Auditory system tends to be a little bit faster in humans. Some of these, since these are mostly sensory peaks, they don't have an experimental manipulation per se, but they do have very nice max scalp amplitudes. So when you look at the scalp propagation around that peak, you see that the maximal peak is anterior for auditory and occipital for visual. Makes perfect sense. And we have interpretations and sources. As we move to peaks that tend to be later on that are thought to be related to cognitive processing, say the N2 or the N170 has been identified for face processing, so your standard face versus houses. Whenever you see the face, you get this N170 negative peak around 170 milliseconds. When you see houses, you don't always get that. And when you see sort of unrelated stimuli, you definitely don't get that. It's also been shown slightly later for a go, no-go, and whatnot. And as I said, N400 before and then P600. Wow, I even did extra slides on this one. So as you've gotten the sense, one of the major measures that we have for EEG and ERP is the amplitude and latency. So we can just measure the amplitude in microvolts and the latency in milliseconds for each of these peaks and say, if we have different conditions, here I have rhyme and non-rhyme conditions, and I have left and right hemispheres. Here I'm comparing, this was work coming out of my dissertation, comparing children who went through a reading intervention, and you can see that children who were typically developing and children who had responded to a reading intervention show fairly similar P1, N2, P3 responses, but then the children who didn't respond to the reading intervention show a fairly different, this dotted black line response. And so oftentimes we'll take these numbers, amplitude and latency, put them into our favorite statistical program and just do standard t-tests or ANOVAs or whatnot. But there are some caveats to all of these measures that I'm going to talk about. For instance, there are variations in the ERP trials. So what I showed you before was the average of, say, 40 trials. But here, if I look at variation of amplitude and latency in both monkey and man, over 100 trials, I take 10 of them and I average them together, you can see that there's a lot similar in terms of this peak structure, but there's a lot of variability that's not necessarily being captured by these grand averages of an individual, both in the monkey and in the man. But fortunately, a lot of this variability does sort of lead towards averaging isn't a terrible thing to do, but a lot of the field is now moving towards examining these individual trials or even subsets of trials. We also see variations in the peaks, just to sort of drive the point home, that the peak may land at slightly different milliseconds over time, and if we have more variation, then we oftentimes get a wider peak, or sometimes we even get this sort of bimodal peak. And this is often an indicator that either we're seeing some really interesting cognitive process, or sometimes that your equipment isn't quite working right. So say the timing of your experiment is off. And this was, I can't tell you the number of times that we've sort of seen this in an early subject and then said, oh wait, something's going wonky about our timing. Less variation in the peaks obviously gives us sharper peaks, but it also gives us higher peaks. And if we are to look at sort of the number of trials that we put into, you can see that the more trials that we average together, is the smaller the ERPs. So here I have, say, 16 trials or 32 trials, 48 trials, and 64 trials. The more trials going in just means the smaller, because we get a little bit more variation, and that variation ends up reducing the overall amplitude. There was a formula at one point that was proposed that the signal-to-noise ratio in ERPs is 1 divided by the square root of the number of trials. So say you have 25 trials, that means that 1/5 of your signal is noise. And for a lot of people, they took that to mean that they should present hundreds, if not thousands, of trials, which meant that their participants were sitting there with electrodes on their head for hours and hours on end. It turns out that there is sort of a sweet spot that, in this case, my dad came up with, which is right around 25 trials. We start to see the trade-off between ERP signal improvement and sort of the number of trials going in. So you don't have to have your participants staying there for hours. In fact, we know based on split half comparisons where we've taken the first, say, 20 trials and the last 20 trials, that the ERPs that you get at the beginning of an experiment are fairly different from the ERPs that you get at the end of the experiment, which really begs the question of, if you're doing hundreds of trials, what are you really getting at the end of the day? In addition to peak amplitude and latency, we have a number of other metrics that we can record. So scalp topography came about with the advent of multi-channel recording. So if I have 256 channels over the head, I can, at any given time and point, say this is where there's, say, positive activation, and here's where there's negative activation in terms of the dipoles across the scalp, and then I can compare my conditions like this. So this was a rock, paper, scissors game where they you could either win, draw, or lose. I'm pretty sure everyone in here knows how to play rock, paper, scissors. Here we were a little sneaky in that the computer always came out with roughly even numbers. In addition to scalp topography, we also have frequency analysis, which was in many ways subverted by wavelets, which allow you to do frequency analysis, or FFTs, while keeping a lot of the temporal information. And then the sort of, I'm going to put it in air quotes that you can't see on the recording, the new kid on the block, source analysis, allows us to have pretty brain pictures that make people feel good inside and make it also be comparable to fMRI in many ways. Now, there's two fairly important things to keep in mind with EEG recording, and one is called the temporal Nyquist, which is that if our signal is something like this, being recorded at 100 hertz, and you can see that it's bouncing around a fair amount, if we collect that at a slower frequency, say 50 hertz, you lose a little bit of the detail, and at 20 hertz, we start to lose a lot more of the detail. You can see that we're still getting most of the main peaks, but we are losing a lot of this variability from moment to moment. And then if we get down to 10 hertz, then we lose even more. Now, I'm not saying this because you're going to be like, oh, who would record at 10 hertz? Like back in the day when we had hard drives that were 256K, maybe that was something to worry about. Well, it turns out that auditory brainstem recordings, like we need to start sampling at 8,000 to 16,000 samples. And if you don't have that sampling rate, you can't pick up on these very subtle brainstem responses. So it's important to be sampling at 2 to 4 times, if not more, the frequency of the peaks or features that you're trying to sample. So also important is that this gets represented in our fast Fourier transform FFT. So if this is a composite of sine waves, 6.5, 10 hertz, and 19 hertz, you can see that at 100 hertz recording, I separate out those 6.5, 10, and 19. At 50, I continue to pull out almost exactly the same amount of information in terms of our FFT power. But as soon as I go down to, say, 20 hertz or even 10 hertz, you can see that we're starting to lose some of the characterization of these frequencies. Also important is this idea that there's a spatial Nyquist. So say there are a whole lot of features going on in the head. If you don't have an adequate number of sensors to be able to measure this, you will get a fairly fuzzy picture. And so if we go from 129 electrodes, where we could pick up something like this, to 64 to 32 to 19, you can see that we're really starting to lose a lot of the features spatially that could be captured. And a lot of this information may be the differentiation between clinical groups, or it could be the differentiation between your experimental conditions. It also adds complexity and lots of fun with statistics. Of course, there is a link between amplitude and the topography. So as I showed these before, this is research identifying differences between children who were bottle-fed versus breastfed. And that is that, can sort of skip to the conclusion that we found that babies who were breastfed versus formula showed faster ERPs and also seemed to be a little bit ahead in development in terms of their ERP than babies who were fed formula. But what I really want to draw your attention to here is that these are just slices in time of the scalp topography. And so I get one of these scalp topographies for every, say, 4 milliseconds or 1 millisecond, depending on how quickly we're sampling the data. So there's a huge amount of information here that we've only begun to scratch the surface of. We're going to chalk this up as one of the things that not a lot of people are doing with their data that could be done. To show you why this is important, here is the scalp topography of an infant hearing the speech sound ba, and you can see that there's sort of initial activation cropping up here in sort of the middle frontal area, and then that goes away. And now we get some activation in the sort of left occipital, temporal areas, and that kind of hangs out for a while, and that's it. So we get these nice discrete activations in the babies hearing the sounds. And now I'm going to show you what it looks like in the adult brain, which this is about a second, but I've slowed it down to be whatever, 10 seconds. Yeah. 20 seconds, so. But it's representing a 1 second after the peak. So here's an adult, and you can sort of see how quickly the activation is sort of swooshing all over. And you can be fairly confident that once we hit about 600 milliseconds or so, the person is thinking about their grocery list and what they're going to do with the $40 that you paid them to be in this experiment. But it just goes to show you the contrast in the amount of temporal information that can be captured with plenty of electrodes in EEG. Whereas in fMRI, we sort of average over 8 to 12 seconds. So this is the stuff that's going on while the bold response is happening. So I mentioned frequency analysis is another great way to go. So we have our ongoing EEG, and if we do fast Fourier transforms, this gets decomposed into different frequency bands that people have named for us. So delta 0 to 4 hertz, theta 4 to 7, alpha 8 to 11, sensory motor rhythms, beta, and gamma. And many of these rhythms are related to different clinical disorders or to certain cognitive processes. So I'm just going to call one out here, which is we see boosts in theta activity while people are doing complex decision tasks, whether that's gambling or whether that's risk aversion tasks. And we see this modulated by people who are high in anxiety or low in anxiety. And this is work that's continuing to be done by Mike Crowley, who's at the Yale Child Study Center. Now I'm going to talk about source analysis because, well, it's cool, and also it relates to much of the rest of this summer course, which is very fMRI focused. So just to give you a little bit of terminology, there is a forward problem and an inverse problem. The forward problem is that given the sources in the brain and some kind of mathematical propagation model, we can say what will the scalp topography look like, or topology. The inverse problem is harder. The inverse problem is saying, given a particular scalp topography and your propagation model, what are the generators of that? And why this is hard is because we don't actually know what the propagation model is. We can come close, or we can get somewhat convincing ideas of this, but in many cases, this is like trying to tell where lightning is going to strike if I constrain the model in a particular way, or if I send someone out into the middle of a golf course holding a very large piece of metal over their head. So in order to try to do the best source analysis that we can, we really need a number of things. So I think I can usually sell people on the idea that an adequate number of sensors given the spatial Nyquist is a good thing. And here we have 256 electrodes. There were, not too long ago, there was a time when people were saying if you need more than 8 channels, then you must be doing something wrong. And I think that a considerable amount of research has now shown that indeed more sensors is better. We need to know the geometry of the head and where the sensors are on the head, and we need to be able to build an accurate head model. And oftentimes this means getting an anatomical MRI on your participants. We need to know the conductivity of the head, and there's some really exciting research going on about this where we actually do current injection into the head, and then we can see how much current goes through the head versus along the scalp. And I usually like to call this sort of like electronic sonar. So you're basically using these resistance bands to figure out like where electrical current flows freely and where it doesn't. And then once we have all of this information, oftentimes we make do with some kind of propagation model. The very earliest head models were things like this. This is the Sun-Stokes model, where we just have concentric circles with different mathematical properties or different resistances. So we have the brain, we have CSF, we have the skull, and we have the scalp. And we believe that the electrical current coming out of the brain is distorted in different ways from each of these concentric circles. Anyone want to raise their hand and say that's exactly how it works? Oftentimes, this was the best that we could do, particularly with computational resources as people started to do source analysis. But nevertheless, it has evolved to one of the more complex models available today, which is the finite difference model, which gives us not only the geometry of the brain, which is not regular and definitely not a circle, but also different conductivity values for each type of tissue in the brain, say white matter, gray matter, CSF, ventricles, skull, scalp, eyeballs, things like that. And just to sort of compare a series of these, so that spherical shell model is what I showed you first. It's great for fast calculation, but as I said, it ignores our head shape and the complex geometry. There's a boundary element method, which is great if you want to do surfaces or cortical sources. And there are plenty of people who continue to argue that the EEG or ERPs that you're measuring at the scalp are almost entirely caused or generated by cortical sources. And so many of the models, whether you're doing this in, say, Brainstorm or M&E or Field Trip, oftentimes will allow you to make this restriction, and boundary element models are great for this. But if we want to look at volume representations and the subcortical activators, we have two choices. One is the finite element method, and one is the finite difference method. And both of these are great for volumes. The FEM is slightly easier to calculate, whereas the FDM is more complex. And what makes the FDM more complex is that we get point-to-point changes in the current structure over time. So it will do sort of this autoregressive modeling. And if you're interested in this, I have a very large stack of papers on my desk. Many of them say they haven't been read yet, but I have some great reviews as well. For a long time, people made sort of this distinction between two different kinds of source analyses. One is called the equivalent current dipole, or ECD, and the other one is called the distributed linear inverse, which was sort of popularized by this method called Loretta, and then we had Laura after that, and then we had S. Loretta, which were just basically different ways that we constrained the electrical currents. I will say, before I get too far into this, Loretta tends to give us incredibly believable results. So if we present things that we see in fMRI in EEG and then we do source analysis, we oftentimes get overlapping sources in the EEG that we do in the fMRI. But this is also based on an idea that in Loretta, all of the voxels, Loretta and for that matter, like all of these minimum norm estimates, is based on this idea that all voxels in the head are active to some amount at any given point. And we know that that's not actually how the brain works. We know that we have discrete firing of neurons, but nonetheless, these minimum norm solutions tend to work fairly well. So this is one of those, the math works well, but we're probably only approximating what's really going on, and we're probably missing a lot of things. So the equivalent current dipoles assume a very small number of sources, say like 2 or 4 sources in the brain. We're pretty sure that you're generally looking for more than that. And what it does is we plop some dipoles in the brain, and then we iteratively search for the location. And we can do this with PCA. The computer can kind of figure out where it thinks those dipoles should go. But also, people are often fairly good at saying, hey, this is an auditory task. And if I look at my N100, I know that's supposed to be auditory cortex. Why don't I put the dipoles in auditory cortex? And it'll find them fairly quickly. It's like the keys are always in the last place you look. If you put your keys in the same place, then the last place you look is the first place you look. The challenges of equivalent current dipoles are that we need this high user involvement, which often means low replication. It's gotten better with these automated methods, but still, when we were doing equivalent current dipoles, we were seeing replication below 50% into trained postbacks or even postdocs. And then we also have to balance that time window, the number of dipoles, and the orientation of the dipoles, because again, we have, say, 1,000 data points over a 1-second period. And as I mentioned, distributed linear interverse, often synonymous with minimum norm. And again, all dipoles are active in the brain to a different degree. And it's also an underdetermined model, which just means that we have many more voxels than we have data points going into it. So sort of the best we can do. There are no true or unique solutions in minimum norm, so we need to apply some kind of constraint. Minimum norm just says we're going to take the smallest overall intensity of activation over all the voxels in the head. We can also do a weighted minimum norm where we, say, apply weights to compensate for superficial sources. This is a fancy way of doing smoothing. Or we can do regularization, which sort of reduces the effect of random noise or sometimes subcortical structures if you don't think that you can get subcortical activation. Over the years, we've kind of come up with this idea that we have good, better, and best, if you feel like you're buying a car. So if you want to do source analysis, the best solution is to have an MRI on your individual subjects and the EEG electrode locations, and I can show you how we get those. And then obviously you need the EEG to do source analysis. And this combination of MRI, electrode locations, and EEG allows us to build individualized head models for each of our participants, so we can actually do person-specific EEG source analysis. The next best thing is to have the electrode locations and the EEG data, but no MRI, in which case we can do some interesting warping to try to make template head models. And this is actually fairly common. We used to do this in MEG as well when we didn't have an MRI on everyone. And then finally, sort of the OK one, do at your own risk is I only have the EEG, and I'm going to assume some default sensor locations and some default MRI space. And don't do this last one, if you can help it. Electrode locations are actually fun to gather. So we have this dome, or actually over in Building 10, we have a handheld sensor. And we put someone like me into this contraption that looks like Darth Vader's little private chamber. And it has 11 cameras on it that take pictures of the person wearing all the sensors at all the different angles. And then we're able to build a 3D model representing all of those channels over the head. And then we're able to use that information not just for source analysis, but also for, say, interpolating bad electrodes. So if this electrode is bad and I know the distance between this electrode and the surrounding electrodes, I can do a weighted average with the closer electrodes contributing more to interpolate that data. Okay. I'm going to talk about EEG and fMRI gain similar results and also MEG. I'm going to start this off by saying there is limited research covering language in simultaneous EEG fMRI. In fact, the field of simultaneous EEG fMRI is still fairly young because of artifacts that I'm going to tell you about in a little bit. So if you're looking to make your impact or mark on the field, this is a great way to do it because we have the equipment. It is approved on many protocols already, and people like wearing the net. It makes for great photos on Facebook for them or whatnot. Nonetheless, there is extensive research in both fMRI and EEG showing that there are differences between typically developing individuals and individuals who have reading disabilities or dyslexia. There are changes in the brain with development of language that are unique and different from just changes in the brain related to age. And there are changes in the brain following a reading intervention. So if you have a student who is struggling in schools and you put them into an after-school reading program for as short as, say, 30 minutes a day, and they start to respond to this reading intervention, we see nice pre-post differences that are not there for individuals who, say, just continue on struggling. And there's converging findings across these different neuroimaging techniques that I'm going to show you. So for instance, ERPs of reading groups. So here's a study with 27 children, 9 to 12 years of age, and the ERP amplitudes and peak latencies are shown to decrease as our reading skills increase. I'm not sure what happened with the typo, maybe a font issue. But as you can see, if we look at the number of correct for word across our three groups, so we have our above average, and our below average readers, you can see that everyone's pretty good for words because sight word reading is a fairly strong or easy skill to develop. Part of the reason that sight word reading was so popular in schools until it was discovered that phonics was better. But then if we go into the it's not a word but it sounds like a word, we get nice differentiation between our groups. So our above average readers continue to do well, whereas our average and below average readers don't do as well. And then if we present non-words and have them sound those out, or even just recognize them as not a word, you can see that, again, we get differentiation between the three groups. The major difference that we showed here is that there are hemisphere differences with increased reading skills. So the better reader that you are, the larger the amplitude of the left hemisphere reading circuit. So say over IFG and temporal parietal. And this is pretty consistent, so I want you to hold on to this idea that the left hemisphere showing more activation than the right hemisphere in more proficient reading skills. This has also been shown in MEG. This is work coming out of Akasimis and Andy Papanicolaou's lab. Andy's now in Tennessee, but this was collected at TIR at University of Texas Houston Medical. So if we have people listening to words, you can see that there's a decent amount of activation in the left hemisphere and some activation, some dipoles appearing in the right hemisphere. In the impaired readers, the non-impaired readers tend to show less activation in the right hemisphere and a little bit less activation in the left hemisphere with a little bit more distribution. And I've just highlighted in this square, or rather Andy highlighted in the square, this sort of temporal parietal area. If we then look at reading words, you can really see that there's quite a difference between impaired readers, where we see almost no activation on the left hemisphere and a whole lot of right hemisphere activation in this temporal parietal area. And then if we look at the non-impaired readers, it's the opposite pattern. So nice, concise activation in the left hemisphere temporal parietal and not very much activation in the right hemisphere temporal parietal. This has been replicated also in fMRI. So Temple in 2001 showed that essentially if we look at differences between reading disabled and and typically developing individuals, we see that the normal reading children show activation in, say, IFG and temporal parietal, and then the dyslexic children show less activation in IFG and no activation in temporal parietal, while continuing to show considerably more right hemisphere activation. Ben and Sally Shaywitz, along with Ken Pugh at Yale and Haskins, showed that if you are to correlate the activation in the visual word form area with their Woodcock-Johnson word attack, your ability to read non-words that sound like words or are sound outable, you see a nice correlation, which is the more activation in the visual word form area is correlated with your ability to do this non-word reading task. Going to shift a little bit and talk about intervention studies. So we're going to be talking mostly about changes in the reading circuit associated with your improvement in reading ability. So again, here's the follow-up from Temple's 2001, came out in 2003. They showed that children with no remediation, so normal reading children while doing a rhyming task show activation in this IFG and temporal parietal region. The dyslexic children while rhyming before the task show no activation, in this case in the temporal parietal or IFG. But the dyslexic children after remediation show activation in both this IFG and temporal parietal areas, which is really, really cool. But the other thing that's unique is that these dyslexic children, in addition to showing sort of this normalization trend of activation in these areas, also show increased activation in the right hemisphere associated with them becoming more proficient readers. So there's sort of a compensatory mechanism kicking in, which is basically they're scaffolding on more brain tissue to do the same task that their typically developing peers were doing. And if we just look at the total change in language score versus the change in this temporal parietal area, you can see that there's a decent correlation between how much improvement we see in reading and how much activation there is. Ben and Sally Shaywitz with Benita Blackman's intervention, again at Yale and Haskins, showed that if we just contrast year 3 and year 1, we see that after an intervention, we see considerably more activation in IFG and temporal parietal and also visual word form area, so these 3 parts of the reading circuit. Now, what is not on the slide that is just incredible is that Benita Blackman went and found these children 10 years after going through the reading intervention and was able to show that they had sustained improvements in their reading from having only participated in this reading intervention for a fairly short period of time. I think it was a 1-year intervention. So every little bit counts in terms of becoming a more proficient reader. And this work has also been replicated by people like Hugh Katz and Chris Lonigan in terms of the benefits of after-school reading programs, particularly for struggling readers. And then, of course, the MEG results are similar. Again, Akas Simos and Andy Papanikolaou in 2003. Here are the pre-brains, and here are the post-brains on the right. And at the time that this paper came out, we really didn't have a great way of representing how this should look statistically. And so the decision was made to just print all of the different brains. So you can see pretty much across the board in the left hemisphere, not a whole lot of activation. We have a couple of individuals who show a decent amount of left hemisphere activation. But in the right hemisphere, we see a lot of activation before the reading intervention. Switching after the reading intervention, we see a considerable shift in activation. So now we're seeing a lot more left hemisphere activation, particularly in this temporal parietal region, and a decrease, for the most part, in the right hemisphere activation. And we like to call this, or I like to call this, the intraocular traumatic test of significance. That's when the results are so obvious that they leap off the page and hit you between the eyes, and you have that aha moment. And then here's the ERP response, again coming off of my dissertation, where we showed that following a reading intervention, we see pretty distinct differences in children's abilities to differentiate rhyme and non-rhyme stimuli, where the typically developing children and the responders to the intervention show a particular pattern, whereas the non-responders tend to show sort of this decreased activation in their ERPs. Now, what was really particularly interesting here is, if I do say so myself, is that we actually see 3 group differentiation, particularly on the non-rhyming stimuli, where you actually can see that the typically developing children are different from the responders and are also different from the non-responders. And what's incredible here is that the typically developing children and the responders read at basically the same grade level. Actually, in this case, the responders showed a slight uptick in reading compared to the typically developing children. And what I really like about this, or what I want to showcase in terms of the importance of EEG, is that this distinction is made between responders or non-responders, or between children who are reading at grade level and children who are not, happens as early as about 200 milliseconds after they see the word. So incredibly quickly. And in many cases, the latency is the thing that differentiates different types of clinical disorders, whether it's ADHD, whether it's autism spectrum, whether it's reading disabilities. And it just highlights the importance of gathering a measure like EEG. Now, if I want to try to differentiate them using, say, very, very basic machine learning, like a discriminant function or supervised learning, you can see that the classification accuracies are fairly good. But if I just say, who are the children reading at grade level and the children not reading at grade level, I get a 96% classification accuracy, which I think is fairly good. In fact, this is not the first type of paper that has tried to do reading classification. My dad's paper in 2000 came out showing that they could identify at birth children who would struggle with reading at 8 years of age with about an 89% accuracy. And in a previous talk that I gave, I got to say, take that, Dad, and he called me about 5 minutes after that and said now his model's up to 98% classification accuracy. So it's an ongoing feud. We have lots of things to talk about over Thanksgiving. Okay, so general themes coming off of this. There are activation differences between good and poor readers, whether it's MEG, fMRI, or EEG, ERP. We are seeing modulation of different parts of the reading circuit. We're seeing laterality differences. Left hemisphere tends to be more strongly activated in good readers and less activated in poor readers. And we see that our reading interventions across the board, no matter what kind of reading intervention, as long as it's a useful reading intervention, as long as it gets results, tends to show a normalization of the reading circuit, so more activation in those reading areas. And in some cases, we also see these compensatory activations, so recruitment of right hemisphere or other cortical and subcortical areas to perform the same types of reading tasks. So let me shift gears now for the thing that everyone's been waiting for in the last, say, 12 minutes of the talk. So there is quite a bit of information that can be gathered from separate sessions of EEG and fMRI. In fact, EEG, fMRI simultaneously does present challenges with artifacts that I'll talk about in a moment, but the incredible power of doing these two together is that we're getting the same environment and the same time courses. So we can actually link trial-by-trial activation in fMRI to the EEG ERP responses. And we can look at individual trial modulation, and we can also look at, say, coherence or resting state relationships at the same time. And I also want to really highlight the same environment thing. So oftentimes, EEG experiments are done in a lazy boy recliner or a very comfortable chair looking at a monitor in a peaceful room with posters on the wall. You feel very at home. And I don't know about any of you, but I don't tend to feel very at home in a 60 centimeter bore of an fMRI laying on my back with loud noises. So in order to really make those data comparable, doing simultaneous EEG fMRI allows you to take in those nuances of being in this fairly restricted environment. So I said there are a couple of artifacts that we really need to keep in mind. One is the MR artifact, or the gradient artifact, which gets represented like this. And the other is the blistocardiogram, or BCG, which is caused by movements of the electrodes, typically on what I'm going to call the fleshy part of your face, every time your heart beats. Because what happens is your heart beats, and those electrodes move just a tiny bit in the magnetic field, and you get current induction. Now, it turns out that these are fairly, well, one of these is really easy to deal with, the gradient artifacts, because what we can do is we have the marker for the TR, the time to repetition, and we can just build a template of all of the gradient artifacts over the entire EEG session, and we can take that template and we can subtract it out, and we go from something like this with the raw signal to something like this where we have our artifacts removed. The BCG is a little bit harder to deal with because it is most prominent over the facial electrodes. But we do know that it follows the heartbeat by approximately 250 milliseconds. And we have an EKG channel that tells us when your heart's beating. So we have a linkage, a pure linkage between like, hey, the heartbeat right around 250 milliseconds, we should be on the lookout for this BCG. And then we can mark it and model it with a PCA. And one of the nice things about PCA and ICA for that matter is that you can take out the component that is causing havoc and then reconstruct the signal without it. And a lot of people have shown that PCA is sort of the way to go for this. That said, it does end up being slightly messier EEG than EEG collected outside the scanner, and so you might want to add a few trials here and there to try to make up for that. Don't go crazy. We don't need 2 hours of scanning. Now, here's a perfect example of sort of classic ways of doing simultaneous EEG fMRI, and I will say this is not a criticism at all of this paper. This is Debner, 2005, and this is an awesome study. So this is a flanker task, so we have arrows going in a particular direction, and then we have a middle arrow that appears to either go with the flow or against the flow, and you're, as the participant, only required to identify where that arrow is pointing. So is it pointing left, click left, is it pointing right, click right. And what they did was they analyzed their data and found that they got a particular scalp distribution for at about 250 milliseconds that looked like this. And then they did source analysis, and they found out that their main equivalent current dipole is in the anterior cingulate. And then when they went over to their fMRI results, they found exactly the same thing, so this activation in anterior cingulate, which shows a nice linkage between the EEG and the fMRI. We did the same experiment, we did source analysis on both, and lo and behold, we find the same activation. And it's true that this is actually how most source analysis is done in the field right now. So most people collect their EEG data, which again is the hardest part of any study. They grand average all their data together, so they take all their participants and they combine them together in one waveform. they pretend all their subjects have the same brain, the same electrode coordinates, the same everything. They plot out the sources on the brain using some minimum norm method, and they declare victory and they publish a paper. So, and again, sources to scalp topography to measured by electrodes to waveforms. And the way that a lot of people are starting to look at solving this is by going to source space. So there are a series of products or free softwares out there, M&E from the MGH group, Field Trip and Brainstorm. M&E is in Python, and then Field Trip and Brainstorm are both in MATLAB. What they do is they go immediately to source space. And the nice part about this is that we're then able to overlay our EEG and our fMRI results on the same participants and say, where do they agree and where do they disagree? Or you can even go one step further and start to say, okay, I get a source here. What does the EEG or ERP look like for this particular source coming out of this particular area of interest that differentiates, say, my different clinical groups? The disadvantage is that, again, even though we're going in source space, we have this temporal domain. So how do we deal with the fact that I have 1,000 time points that all give me slightly different sources? And how do I pick the right one? So here's an example. This is EEG and fMRI source analysis on the same individual. But you can see here that the scalp topography is changing all the time for this particular experiment. So there's a standard P300 parahippocampal activation. And what I did was, because I knew it was a P300, I picked a particular point in the P300 that was pointed out to me by a temporal spatial PCA. And then I did source analysis on that. And you can see that there's a lot of similarity between the EEG source analysis and the fMRI. But there's also some differences in that we get considerable activation in parts of the EEG source analysis that aren't present in the fMRI. And the old way of thinking about this would be to say, oh, whoa, EEG is getting something wrong. But I think that the important way of looking at this is that maybe fMRI is missing something because we have a whole lot more time points on the EEG that might be informing these sources. And so there are ways of dealing with this, including that you can constrain your EEG solutions by your fMRI, or we could say try constraining our fMRI by our EEG. Both of those have been published at least a couple of times. But one of the things that I really like to do, and that I sort of alluded to earlier, is say in an N170 experiment with faces versus Chinese characters, you can see that EEG source analysis does give us this nice representation where faces are more prominent in the right hemisphere fusiform area and characters are more prominent in the left hemisphere fusiform area. And if I plot out the left fusiform area activation, particularly, if I plot out the left hemisphere fusiform activation, and I do that by finding the sensors that are most representing this source part of the solution, you can see that I get a nice differentiation where indeed characters show a larger N170 than faces, and then I also get the rest of the waveforms that you could do any number of other statistical measures on. Ooh, that was a mouthful, and 4 minutes to spare. So with that, if you have any questions, I'd be happy to take them, and you're also welcome to email me at any point. Any questions? Guys look like it's 3 o'clock or 4 o'clock on a Wednesday afternoon. Okay, well, I'll let you out of here. Thanks.